Our speaker this afternoon is Professor Brent Christner, preeminent scholar in the UF Department of Microbiology and Cell Science. He supervises the Christner Research Group, which uses a combination of laboratory and field approaches to study microbes and their processes in glaciers and ice sheets, karst aquifers, and the high atmosphere. This research has involved more than a dozen expeditions to Greenland and Antarctica, with emphasis on microbiological processes that control the flux of nutrients and greenhouse gases to the ocean and atmosphere. Professor Christner also has interests in icy systems beyond Earth and is the lead principal investigator of a NASA project to better understand how the signatures of life can survive under the extreme conditions present on the surfaces of ice-covered moons, such as Jupiter's moon, Europa. He is currently in Austin, Texas, concluding an experiment to study biosignature preservation under space-like conditions and how that connects to what we may learn from the Europa Clipper mission. Professor Christner received his Ph.D. in microbiology at Ohio State University. Searching for life in the icy subsurface oceans of alien words. Please welcome Professor Christner.
Well, good afternoon, everybody. And I do apologize that I'm not able to be there and give you this talk in person. But I have good reasons for not being there, and I think I can add a little bit to my talk by giving you some very new data from some of the efforts that we're undergoing. As you heard in my introduction, my interests range in things that are microbial, and in particular, things that are microbial that inhabit places like the polar regions and ice sheets of Earth, environments that are deep underneath the surface, as well as places where microbes subsist high in our atmosphere. These all seem topics disparate in nature, but they all connect really in through one way, and in one word, they all connect through their relevance to astrobiology, and that is something I'm going to tell you a little bit more today. Now, I know in the last few weeks, you've heard from a colleague of mine, Amy Williams, talking to you about efforts to look on Mars for evidence of life, and Amy may disagree with me, but I will tell you that the best place we should be looking for life in our own solar system is not Mars. It's actually deeper in the solar system, in places that we know have oceans. I'm showing you on my title slide right here 2 examples of what now tally about a dozen places in our solar system that we refer to as ocean worlds. That means they have a vast ocean that isn't visible. As you'll note, you're looking at on the left here, this is a moon of Jupiter called Europa, and this is a moon of Saturn called Enceladus. You don't obviously look at these bodies and see an ocean, but if you could go tens of kilometers underneath the surface, we now know that these places have vast quantities of water and oceans that rival the Earth's ocean. I do want to point something out before I move on from this slide, is that I'm showing both Europa and Enceladus like they are equivalent in size. And just to give you some sense of scale here, Europa here, its surface area is about 3 times the area of the US. Whereas in the case of Enceladus, it's a much smaller body. Its surface area is about the size of Texas, the state I'm in right now. To sort of set you up for the rest of the talk, I want to just sort of fill you in that in the last few decades, New data on these and other planetary moons have really changed our conceptual understanding of what might constitute a habitable and sustainable biosphere. See here, let me get some of these off of here. Nope, I'm not doing that. Okay. So if you're a product of the 20th century, then you likely learned that probably the only places off Earth we might find life is Mars. And that's sort of reflected here in this excerpt I pulled from a textbook in the 50s, right? This is a textbook that was published even 20 years before I was born. But the message to my generation was the same. If you're thinking about the possibility of life deep in our solar system, it didn't make sense to us at that time. We just didn't understand, first of all, how robust life was. And we also didn't really understand the possibility that there could be life many kilometers, not only oceans, kilometers underneath the surface of these worlds, but life there is as well. By extension, this view was transposed on our view of the universe, right? That where we estimate there might be 10 to the 23 of these planetary systems. And what you see here is this slide is showing you some results from a paper that was published in the late 90s. Just for some frame of reference on my career, this was the second year of my graduate work in which I was very excited about because I was studying deep ice cores, studying how these rarefied environments underneath the Arctic ice sheet could potentially hold these interesting ecosystems. Little did I know that the implications of this research were much broader because, again, very early in my graduate career, a paper was published, and this is based on observations made by a mission called Galileo, which was a Jupiter mission. And the authors on this paper were part of the imagery team for Galileo. And I'm showing you an example of some of the images and data that were collected from that mission. What you see here are, I could actually show you pictures of sea ice, where sea ice breaks together and refreezes, that I could switch the images and you would not be able to tell whether or not that was ice from the surface of Europa or from the Arctic. These kinds of images, these can almost be, if you look carefully, you can almost put these back together like puzzle pieces. And there is no real way to explain how this could form without the possibility that that there's liquid underneath and that these are actually moving and refreezing. And this actually was the seed of a concept by which we now understand to be not just the case on Europa, but the case for multiple other worlds in our solar system, where in fact these very icy shells we see on these worlds are underlain by, in some cases, oceans that are very deep. We don't really know how deep this ocean is yet, many kilometers underneath the ice surface. And how do these oceans stay liquid? The Carnot hypothesized that because this is a moon that's orbiting Jupiter, and that it's actually the tidal flexing that's induced by Jupiter that is keeping this liquid. That's going to advance my, is that advancing my, there we go. All right. And the year before the paper I just showed you was actually published, a mission was launched called Cassini. It was actually the Cassini-Huygens mission. This was a mission to Saturn, and part of what it was going to do is study Saturn and its rings and its moons as well, with a particular emphasis on Titan. It actually had a probe that descended to the surface of Titan and sent back data to Earth, and was the first time we've actually landed on one of these planetary moons. But at any rate, when it arrived in the Saturn system, it started imaging Enceladus. And what it discovered when it imaged its southern pole are these jets, these plumes of icy material that are actually ejected from Enceladus into space. Remarkably, this Cassini spacecraft was equipped with 2 crude mass spectrometers, and they were able to fly through material that's ejected from these plumes and detect salts and water and organics. And of course, these have actually raised questions about where these organics came from, and if you had the right equipment on the spacecraft that would have traveled through these plumes, could have you actually detected life? Now, this is just sort of a little schematic of where we are in our understanding of ocean worlds. By the way, if you dig a little deeper, you'll find that these ocean worlds come into a few categories. We have potential ocean worlds, places where we think there could be oceans, and where there may or may not be. And there are also places that are confirmed through geophysical observations that were either collected by Cassini or Galileo that have confirmed that these places have oceans. And back to when I learned in high school about Earth and its unique place in our solar system as being a water world, not only do we now realize that Earth doesn't have the only ocean, it doesn't even have the biggest ocean. These X's indicate what we believe the volume. For example, the volume of the ocean is more than 2 times that on Earth. We go to a place like Titan, 10 times the volume of Earth's ocean, even though we probably think the Titan of ocean is not inhabitable. It contains about 25% ammonia, by the way. But in particular, Europa and Enceladus, shown at its sort of true size in comparison, these have been targets that have received a lot of attention. When we think about the possibilities of life, water always comes into, liquid water in particular, comes into the equation. And of course, all life as we know it requires water, but water is not the only thing that you need. You need the chemical elements to actually generate life, to produce a cell. You need the raw materials, and you also need the compounds that drive the chemical processes that life requires. In other words, you need an energy source. And we can invoke scenarios on these ocean worlds whereby these compounds are made available. In terms of the chemical requirements for building life, at least carbon-based life as we know it, we understand that if there are similar water-rock interactions that are occurring in the deep mantle of Europa, which has been well studied in context of the Earth's ocean, that could introduce into the ocean what life needs to build new cells, as well as the chemical energy sources that could be used to fuel life. And just like your car not only needs fuel, it needs oxygen to burn that fuel, so does life need an oxidant to burn through this material. And because there's no photosynthesis occurring, obviously, not just on the surface of Europa, but many kilometers beneath an ice cover, we have to find new sources for oxidants on Europa. And we now understand that there are intense radiation processes that occur on the surface of the ice. And because that ice is so dynamic, that eventually gets turned in the ocean, that those oxidants could be introduced. So as we look at these ocean worlds, this is showing Europa, but this is the case for many of them. There's no reason to look at them and say they're uninhabitable because they don't fulfill the basic requirements of life. And last year, a pioneering mission took off from the Cape that when it arrives into a what I will not call an orbit of Europa, an orbit of Europa would be death for this spacecraft. Apart from the surface of the sun, there is no more intense radiation environment in the solar system. And this is because the magnetosphere of Jupiter acts like a natural particle accelerator. And so the way that you get spacecraft to survive in this environment is if you keep it out of it. You keep it away from it. And so this spacecraft will make these flybys where it comes into the high radiation environment for short periods of time to collect data and then leaves. And in this way, it should over the next decade provide a wealth of information on, for example, just surface features and compositions. At the moment, we have the beautiful images I showed you from Galileo, pioneering at the time. Each one of the pixels in those images represents about 100 meters. And so if we want to do something like land on the surface of that, we don't have the kind of data that will allow us to do that with any confidence. This mission also contains a variety of instruments that could actually provide some indications that Europa has life. This vehicle cannot land on the surface, but I showed you those plumes that are ejecting material from Enceladus. There's actually some evidence that this could be occurring on Europa as well. And it's quite possible that in a few years, that Clipper is able to pass through one of these regions, sniff up some of that material that's been ejected from its surface, pass it into either its mass spec or dust analyzers, and actually identify some compounds that might be the kinds of things you would expect a life to be built of. It also contains the kind of instrumentation that has been used in Antarctica for decades to actually not only map the ice surface properties and characteristics, but the depth of the ice. Ice penetrating radar is going to tell us more than we've ever known about not just the thickness of the ice shell, but its distribution. And it will also tell us, we think that there could actually be places within the ice shell where there are liquid water, so-called lakes that actually persist in the, it will tell us about that as well. If we are going to detect life, the kinds of missions that have been proposed to do that would need to land on the surface. And I was fortunate enough in 2017, I was able to sit on a team, what NASA calls a science definition team. And this is what NASA does before it wants to put a mission forward. A bunch of folks get together and write a 500-page document that would propose a mission detailing weights of payloads and energy budgets and science instruments and all kinds of requirements. This was an incredible exercise to be a part of. And what I can tell you is that we were hopeful that when the decanal survey came out in 2000, that the lander concept would actually be prioritized. And I'm sad to tell you that it wasn't, but there are actually some very good reasons for why it wasn't. I've got a few questions here that are actually sort of big questions that we need to know before we put an expensive lander like this on the surface. For example, how to land on the unknown surface. There's actually some heritage in landing in foreign surfaces. I just mentioned Titan. For decades, we've been putting things on the surface of Mars, right? So this has become old hat of such, but actually there's a big difference here. Both of those places that we've landed on the surface have an atmosphere. And so What that means is you can use the atmosphere to slow down. If you want the spacecraft to not pummel and explode under the surface you're trying to land, you need to slow it down. And that means, in a case like Europa, you have to carry the fuel to slow down. And you also need to figure out a way to land on a surface that doesn't have an atmosphere to do that. And probably a concept like the jet crane, if you're familiar with that on Mars, could do that. So that's an engineering challenge. Where to land? If you'll see in this artist's rendition of where this lander is laid down, it's this nice little flat area. It didn't land on something rough where the lander would have fallen over on its top and not been able to communicate or carry out any of its science operations. That would be really bad. And as I mentioned to you, at the moment, we don't actually have the kind of resolution that would allow us to pick a site like that. Oh, that's a perfect site. Like if we'd been 100 meters off from this site, this thing could end up on its top. So this is something that Clipper can supply to us. And here's the third thing that I'm going to address a little more in our presentation. If we actually landed on the surface with the instrumentation to detect the indicators of life, that presumes that the indicators of life, right, which we're not actually saying that there are critters moving around on the surface of Europa. High radiation, temperatures of 100 Kelvin, vacuum in space. These are not the kinds of conditions where life is swimming around and dividing. What we may have instead is a scenario where material from the ocean, which is captured in the ice, the ice shell itself is derived from the ocean. And if the ocean is inhabited and that material gets in the ice, and we can sample that material from the surface, we should be able to find those kinds of things that life would impart to the ocean water. The question remains, is that material that actually is preserved in some state on the surface of Europa that we would be able to detect it? So before I get to that, I just want to draw some connections between sort of the way that astrobiology works for the moment is that we see a place on a foreign world and we use planetary analog, earthly analog research as a way to guide us into not just the technology that we may use, but also the kinds of processes and organisms we might inspect in those environments. And so what I'm showing you here, a comparison on the left and the right, are drawing comparisons to the sub-ice ocean of Europa to sub-ice water bodies that we know about that exist underneath the Antarctic ice sheet. In this case, I'm showing you an example in the schematic of the largest lake in Antarctic, Lake Vostok. Under 4 kilometers of ice is a lake that by volume is the 6th largest freshwater lake on our planet and is about 1,000 meters deep, right? So we know from decades of research on ice cores that have been drilled down from the surface and into this region. This is called accretion ice. What actually is happening here is the ice sheet is traversing across the lake. Water is freezing onto the bottom, accreting on it. And so ice cores that have been drilled into this material have been used to understand processes that are occurring in the lake. And from this, we know that the biology and the chemistry indicative of biology that's in that water column becomes preserved and captured in that ice. So that lends credence to the idea that the same thing could happen on Europa as well. The other thing about these systems is that because in a scenario where we have kilometers of ice, we are talking about a lake that's different from the kinds of lakes that we're all familiar with on the surface, whereby the main source of energy driving processes in those ecosystems is the sun. In the case of these subglacial lakes, the sun is not an option. So we're talking about chemical energy sources that will be driving microbial metabolisms and ecosystem processes. And we know that those occur in subglacial lakes. And again, by extension, they don't make it an impossibility that it could happen in a place like Europa, where there might be where there might be hydrothermal vents like we find in deep parts of the Earth's ocean. And just if you could actually peel up the Antarctic ice sheet, it would reveal that there are hundreds of lakes beneath it. Just Lake Vostok at its position here in East Antarctica is shown for reference. And this is a dated slide. Each one of those little blue dots represents a lake. We now know there's about 700 of these lakes underneath the Antarctic ice sheet. These little lines that you see that look like rivers are actually rivers. These are lakes that have fill drain cycles, and this material is eventually drained to the ocean. I just want to give you a little feel for what it looks like to drill into one of these lakes. This is a drone image from our most recent drilling site. We were there in 2019, and what you see here is about 1 million pounds of equipment. All this had to be traversed about 100 kilometers out onto the ice sheet. And so huge amounts of equipment, and about 40 people were at this site for about a month and a half to drill through the ice, collect material. I'm going to show you a little bit of little data from the system as well, but I just want to point out, if we do this on Europa or Enceladus, we obviously need a completely different model from the way we do this on Earth. And when we were able to get into the lake and pull up water samples, as microbiologists, the first questions that we're asking about are, is there life there? It's obvious to us now that there is, but at the time that this research was done, so it's about a decade ago this work was published, there's actually speculation that these lakes could be sterile. And so, like the images that you're seeing here in A and both B, these are images where we're actually staining the cells with the dye that fluoresces when we expose it to UV light. These were images that were actually generated about an hour after some of the first water samples were generated. These were images that were viewed for the first time with the popping of champagne corks, because not only were we seeing that there were cells in the sample, but you'll see here, every place that there's a little dot, sometimes there's a long filament, that's an individual cell. So not only there were cells, that there's a lot of them. And when you got a lot of cells, that means they got to have something to eat. These, by the way, are just some electron micrographs. These wouldn't be taken on site, but just to show you some more close-up images, these are cells that are quite often associated with minerals. This goop that you see that's encased in the cell is a biofilm. So these are things that cells produce to protect themselves and to attach to substrates. And just because I don't think I've mentioned it, two of the lakes that I've been fortunate enough to be involved in drilling into are in this portion of West Antarctica, and they're under nearly a kilometer, well, one of them is under about a kilometer of ice. In comparison to something like what we might think on Europa, at least 20 kilometers of ice. And cells and the energy currency of life, these are the kinds of things that when we measure in these water samples tell us not only is life is there, but there are processes that are allowing it to generate energy. And in the case of subglacial lakes, dissolved oxygen, DOC is just a fancy way of saying food. These are reduced carbon substrates that the microbes, they can oxidize these for energy using oxygen. And microbes are just simply more physiologically diverse than we are. If there's no oxygen, they can use nitrate, or they can use iron, or they can use manganese, or they can use sulfate. The list goes on and on. And again, back to the kind of infrastructure that's needed to do this. Set to do sediment coring. This is like a 6-ton sediment core that using hydraulics is lower down into the borehole. And when it comes up and it's muddy like this, when we open it, we see things that look like this, right? Those bubbles that are evading from that sediment impart a large portion of that gas is methane. We now understand that the sediments, there are microbes in that sediments that are degrading that organic matter. that is entrapped in the sediment, and they are mineralizing that to compounds like CO2 and methane. That methane, when it diffuses into these aerobic water columns, now mixed with oxidants, serves as an energy source for microbes that live in the water. And from these kinds of studies, we've been able to piece together a conceptual model for how these lakes operate. Again, they're different than the kinds of lakes that we would find on the surface, where in the water column, we would be finding photosynthetic organisms, which would be fixing CO2 and sometimes fixing nitrogen, and then providing that carbon and energy that filters down through the rest of the community. Again, photosynthesis not an option in a subglacial lake scenario. And so the options are chemical energy. And in a subglacial lake, we understand that the sediments are very important for providing the kinds of substrates that are important to biology that get into the water column. In the case of organic matter that is degraded to methane, as I just mentioned, these are processes that also drive nitrogen cycling, as well as mobilization of inorganic compounds from the sediments, some of which life can take advantage and oxidize to generate energy. And we also know that there's some more exotic energy sources in these subglacial lake systems. For example, when you put kilometers of ice over rock and you move it, you crush and pulverize rock. As it turns out, this fracturing of the rock actually creates silica radicals, which when they react with water in inorganic compounds, generate a huge number of compounds, even organics, that life can use. So, from what I've told you about analog scenarios on Earth, we understand that you can put life underneath an ice shell and it can find a way to not only build new cells from the sparse elements and energy that are available, but also potentially thrive. We now understand that if we wanted to go to a place like Europa and Enceladus and detect for life, the go for the jugular mission would be to land on the surface and then deploy some kind of melting vehicle using like a nuclear power source, which in principle could melt down through kilometers of ice, right? And then when we get into the ocean, it becomes an ocean explorer, and then it not only moves around and maps the ocean, but it carries out the complicated task of life detection. Friends, I'm here to tell you we're decades off from that kind of technology being deployed in deep space, which is why in the near future, our best chances to look for life are to look for that evidence that could be preserved in the near surface, that can be accessed with spacecraft and instruments that have heritage. These are things that we can do. Y'all, especially in just hearing Amy's talk in the last few weeks, are probably well versed on what we've been able to accomplish on Mars. It's just amazing that I now sit on graduate student committees that are doing science on Mars. But what I'm showing you here, the latest rover, which took off, it's 5 years ago. In fact, to remind myself, this is a picture of me and my daughter on Cocoa Beach at about 6 a.m. for the takeoff. And it's been there a few years, and my slide is probably not up to date. But since it's been there already, it's traveled around 40 kilometers, surveying, taking samples. It's collected 24 core samples that eventually could be sent back to Earth and analyzed. It sent back hundreds of thousands of high-resolution images. And it could very likely do this for another decade. It's not going to run out of power anytime soon. This information could continue to flow. And I'm here to tell you that the model for scientific exploration on Mars, whereby SUV-sized rovers are able to drive around on the surface conducting science for decades. That isn't going to happen on an ocean world. From being on that science definition team a few years ago, what I learned is that in the best case scenario, what is identified here is the baseline mission. We would be able to land on the surface and collect and analyze 5 samples. 5 samples. And mission durations, not decades. In the best case scenario, the mission could last a month. The reason for this is not lack of technology. The reason for this has to do with the intense radiation environment that's on the surface of Europa. If you land something there, we can't design anything that can survive about longer than 30 days. So the only way you can do that, protected by getting it underground or conduct that mission in a 30-day time frame. Here's one thing. There's you can see a bunch of parameters that are described here. There's one in particular that I want to pay a little attention to. Everybody agrees if you go on the surface, if you go and land on Europa and you would collect material that's on the uppermost surface of the ice, everyone agrees that that would not be the material you would want to get because it has been so excessively processed by radiation that even if it did contain any indicators of life, that those have been erased. And so the report makes a recommendation that to actually get deep enough to get material that has been protected from the space environment, that it would need to penetrate 10 centimeters, 4 inches. Doesn't seem like a big deal. It is a big deal for a spacecraft in deep space to excavate even 10 centimeters. Ice is the kind of thing that I just showed you. On Earth, we can penetrate kilometers of it easy. We're talking about ice that's 100 Kelvin. This ice at this temperature sort of behaves like granite, and that 10 centimeters could be enough, but it could not be enough. And this is actually something that we have to know before we send a mission to do this. And what we don't really have a good understanding of is how these compounds behave under space-like conditions. We don't have a planetary analog for these kinds of environments. And so, for the rest of my talk, I want to tell you a little bit about a project that's trying to help us understand these processes better. This is a project that's funded by a program in NASA called ICAR, Interdisciplinary Collaborations in Astrobiology Research. This is the kind of program that NASA has, the one and only program that is capable of actually tackling a study of this scale. And I'll give you some idea of the scale of that in just a moment. But before I go too far, I just want to introduce the team itself, myself, and many of you already heard from Amy Williams, but we also have from the area, Steve Benner, who is located out in Alachua, as well as Andrea Payne at University of Maryland, Nathan Bremo at Leiden Measurement Technology, Andrew Matadota at NASA Ames, and Bill Stone at Stone Aerospace, where I currently am. So this is a team that's unified in their belief that we can land on the surface of one of these places and be able to detect life if life is inhabiting those oceans. And we are also unified in understanding that this is a problem that we must know more about before we actually try to carry out one of these missions. I think I've probably said this already, but because we don't have planetary analogs that allow us to easily tackle this problem, we don't really understand how when you take a microbe that is in liquid and inject it into cryogenic vacuum, what happens to it? Does the cell explode? Are you able to see any features that would allow you to identify it as a cell? We simply aren't able to do that under conditions on the surface of the Earth. We do have actually very good data on how different types of particle radiation penetrate ice in hard vacuum, but essentially to the micron scale. So when I tell you that there is a requirement to go 10 centimeters in depth under the surface of Europa, no one has actually ever done that experiment. These are based on modeling, because it's actually quite challenging to do these kinds of simulations at scales that are thicker than microns. The other thing that we don't know is that what else is in the ice besides some potential biology and the water, frozen water itself, that matters. And I'm going to show you a little data that shows how the non-ice components of these cryaces could actually have a large factor in their biological preservation. The experiment that I've been in Austin working on since September, and we're less than 2 weeks from actually completing this experiment, is an experiment called Ice Lab. You learn after too long, you get funding from NASA, you need to have acronyms. And so Ice Lab is actually an acronym that describes our experiment fairly well. We're irradiating cryovacuum experiments to study loss and alteration of biosignatures. And in these experiments, we are creating 60 centimeter profiles of ice up to that. Some of these are actually only 20 centimeters. Because composition matters, we're using that as a variable. There are spectral data that give us indications on the composition of surface ice on Europa. As I told you, we actually have collected data from what we think is water that's been ejected from Enceladus's ocean. And so using that data can actually guide us in a way that we can make an ice that we think would mimic that would mimic the surface ice compositions. And we're comparing that to a deionized water end member. And what we're comparing is the survival of a number of potential biosignatures, the kinds of things, if you were going to go and look for molecules, for either life itself or the molecules of life, these would be all things in that category you would look for. Essentially, we're putting bacteria in these samples, the macromolecules, nucleic acids and proteins that life depends on, the components of those macromolecules, such as nucleobases. Steve Benner is actually giving us nucleobases that we might find on Earth, because if we go to Europa, we can't assume that it will have the same molecules that life has. In addition to amino acids and fatty carboxylic acids. And we're exposing these ices to 4 different kinds of ionizing and UV radiation sources. And in these experiments, what we're actually simulating, in some cases, for some of the sources, is a time frame relevant to preservation, to the geological age of these surfaces, although this would be very old for that. But in the case of Europa, as I told you, it's highly intense. We do an experiment for 2 months. We're actually simulating what would happen on the surface of the Europa in weeks or months. Because you would have to add about 3 more zeros to our budget to simulate those kinds of fluxes. Our objective is that when we can actually get dose decay relationships, and we can do these at lower values, we can use them to estimate preservation timeframes over timescales that are relevant for preservation on the surface. And because we don't have samples from Europa and Enceladus yet, as I mentioned, we have to make that. How do we do that? You can't just take the water and throw it in the freezer and freeze it. You don't get a homogeneous mix. It all becomes fractionated. And so what we're doing is we're from ice particles, which we're mixing, as you can see here, in the same kind of mixer you'd use to make a cake. We add in our biosignatures. We dump this into dye, which we use 1,000 psi press to compress it into ice. And because the biosignatures don't play together well, in other words, if there's bacteria in with the organic carbon samples, it messes it up. We have to take single cores that we generate and divide them down the center, and then we recompile them and press them together. That's what it looks like when we press these cores together. And then we insert them into these vacuum-rated nipples, which then can bolt on and allow us to pull down vacuum. These are high vacuum, about 10 to the minus 7 torr, if that means anything to you. Now, we have to then connect these to a manifold, which allows us to not only maintain those pressures, but the sources, which I'm sorry you can't see here, they're above, are actually pointing down. This is our X-ray assembly, by the way. These are inserted into large vats of liquid nitrogen, so we can actually maintain temperatures on the neighborhood of about 80 Kelvin. And then this is what it looks like when they come out, right? Cold. Everybody's got cold hands pulling these things off. And I'm just going to wrap up here by showing you some preliminary data from the work we've done over the last month and a half. And in fact, you are the first audience to actually see these data. Not even all the members of our team have seen them yet. Let me just describe a little bit of what we're doing here. In this case, the biosignature that I'm showing you is the survival of bacteria. And for our experiments, the model organisms that we're using, we're using two because, well, they both have properties that we think make a lot of sense for the kinds of biosignatures that might be surviving in ice in Europa. Maybe not these organisms in particular, but the properties they have would be the kinds of things we might expect in a Europan. Many of you may have heard of Deinococcus radiodurans. It also has the moniker Conan, the bacterium. This is an organism that has extraordinary resistance to both ionizing radiation, UV radiation, oxidants, desiccation, incredible levels of resistance. We put it in this core in addition to an organism that has cold properties. Cyclobacter cryolentis. This is an organism originally isolated from Siberian permafrost. It's an organism that's been demonstrated capable of growth down to minus 10, one of the sort of lowest measured doubling points for a bacterium. So I just want to show you some of these preliminary results, because what you're seeing here, this is depth within the core that we have sampled. In this case, we've exposed this to deep UV. So these are the kinds of wavelengths that you would never really think about on the Earth's surface, because these become filtered out by ozone and oxygen. They're strongly absorbed by them. Part of the reason why we also have to do these experiments under vacuum. But and so there's a lot of biological study of UV, but mainly it's the wavelengths of B and C. What you're seeing here is the log viability. So this is essentially, these are the organisms that remain viable. And the numbers that you're seeing here deep in the core, these are actually concentrations that we put in to start with. And so at these depths, we are inferring that the radiation applied to the surface has had no effect. You'll note here this really jumpy one here. I call these preliminary data because this is a sample we want to redo because obviously we didn't get this one homogeneous, and we know why. Let's put our focus to the surface here. I want to show you the two things that we've recognized so far. These samples that represent the simulant in comparison to water. The presence of salts in these samples has really decreased the viability that we see in these samples. And at least one initial explanation for this is that actually the presence of these salts is likely to be increasing the production of reactive oxygen species, which radiation produces. When we hitting these samples with radiation, most of the sample is water. So that's what most of the radiation hits. Those reactive oxygen species that are generated then react with a compound of interest are referred to as secondary effects. And it would appear that these secondary effects are actually having a detrimental effect to at least viability. And when we are able to do other measurements, when we get back to UF, we'll be able to see if this trend follows with the other biosignatures that we're studying. And there's also a secondary effect that we see. These are data generated by my graduate student. You see she's drawn these lines where she's inferring the maximal extent where viability has been decreased. And what you'll note is that it appears that that depth is deeper for the presence of salts in the Europa Enceladus mixture than the water. Now, this seems to be at odds with what we see on the surface, but what I should mention is that DUV going through ice, it's attenuated exponentially with depth in the ice. So when we get down to these depths, there might be less of the UV that is actually generating reactive oxygen species. But what we do know is that many of these salts, and especially these salts in combination, when we freeze them cryogenically, we're generating salt hydrates. And it's quite possible that these salt hydrates could actually be protecting, acting as like a shield from the UV in this. And this is something we want to follow up on further. Ultimately, what we want to be able to do with these data sets is to fit them into a framework that informs any future effort to look for life on these ocean worlds. And NASA has a way of classifying biosignatures that it refers to as the ladder of life. And I'm actually showing that here. The ladder of life is based on the fact that the lower rungs of this ladder contain the kinds of signatures that if you found, you would be like, that's interesting, but they are not absolute indicators for the presence of life. As you move up the rungs of this ladder, the molecules, like you find amino acids, peptides, even more interesting. But you find an organism that's dividing, that's metabolizing. It's essentially a mic drop. Well, if we think about that ladder of life and the sort of ranking of how confident when we find one of those signatures tells us about what level of confidence tells us that we've actually found life, what I'm showing you here is sort of our overarching concept for the Above and Below project. Like, we're actually arguing that these rungs of life probably respond differentially to, in this case, you're seeing dose of radiation, but we could also replace this x-axis with depth in the ice, right? The data I just showed you, viability, referred to growth and reproduction. And you'll see on this representation that we believe that the signatures that allow growth and reproduction are probably the signatures that are the most sensitive to radiation. So in other words, if we land something on the surface of Europa, it probably doesn't make a lot of sense to try to take some of that ice and put it on a petri dish to see if they would grow. I'm not trying to talk people out of doing that, but as we move down that ladder, right, molecules conferring function, biomolecular components, we believe that these things are actually going to be much more hardy to the effects of radiation. And we're going to test this idea in our experiments. You'll note that there's this yellow line that goes through here. And I just want to explain this. This is, if we run our experiments for 6 months, 6 months straight, that is actually a really long experiment to have an X-ray source on, we could achieve 10 to the 5th gray, right? Uh, Several weeks of exposure on Europa would represent 10 to the fifth gray. We are not able to simulate what is happening on the surface of Europa in the lab. We can't do it. How we are going to inform that is if we can actually generate data, dose-decay relationships, we can use that to extrapolate over time frames, over doses and ages of the ice that would be relevant for the kinds of sites we can encounter on the surface. And I just want to wrap up with a few points that that I mentioned throughout my talk. The place that we want to go, if we want to find the place that is the closest to a sub-ice ocean, would be Antarctica. The lakes that exist underneath the ice sheet, they can serve as models for develop the kind of technology that we might use to eventually enter one of these sub-ice oceans. They also lend credence to the idea that you can isolate biology under ice without light and have a sustainable ecosystem. We've shown that in Antarctica that that's the case, and by extension, that should also be plausible on a place like Europa or Enceladus. Antarctica has also taught us that biology that is occurring in a sub-ice body, that information, those molecules, the cells themselves, actually become entrapped in that ice, which again leads credence to the idea that if we were able to land on the surface of Europa and collect some of that ice, that those indicators would still be there. That is, if we are able to get deep enough to collect material that has not been excessively processed by radiation for millions of years. And that's what the Above and Below project is attempting to do. The next few years, the next decade, is just going to be an incredibly exciting time as data from Clipper, as they start to funnel in, as we start to learn more about its sub-ice ocean, and not only giving us what you actually need to land on the surface, not 100-meter pixel resolution. That would make everyone nervous. We need better than that, and we also need to be able to go to the places that offer the freshest material possible. The freshest material possible would be standing with a bucket right next to the plume. But if that isn't an option, younger is better. I did mention that in the last decadal survey, a lander on the surface of Europa was not prioritized. Part of the reason has to do with the price tag, which is associated with these kinds of flagship missions. From what I can tell, that mission was estimated to cost $5 billion, which probably means that it would cost $10 billion. That's a big chunk out of NASA's budget, especially when you consider what its current state is. These kinds of flagship missions, they come around every decade, right? So if this is not happening this decade, the next decade is really a good possibility for it. And with the data that Clipper would provide, the opportunity is really ripe. I will also mention that there is actually a simpler way. We could try to detect life on these worlds without the need to land on them. Landing on them is a huge deal, not just for all the amount of fuel that you have to carry to slow down. Again, no atmospheres to do that. That's all fuel and weight you got to get off the surface. But you don't have to. You can protect yourself from the radiation environment. There is actually several missions. One, this is an ESA mission proposed, Moonraker, that would do almost what Cassini did. Fly through the plume, collect that plume material from orbit, and try to assess whether or not that material has indicators of life. Whether or not we go to one of these worlds and we either land on the surface or survey the material from orbit, what I can tell you is that when we start those efforts, it represents a new era in astrobiology, and one that I hope I'm still alive to see. So with that, I will stop talking and open up the floor to any questions. I'll try my best to answer them. Everyone's muted, by the way, in case anyone's talking. Maybe I've gave them the whole talk and no one's there.
Well, we have two questions on chat, and we have people in the room. Okay.
I've got a quick question. First of all, I know that the pictures of Europa are color augmented, but there is redness on the surface of that ice. And I wonder if you could comment on what that redness indicates.
Yeah, you are absolutely right. Any images I showed, any images that you'll see if you Google Europa, those are false colored images. Because you mentioned it, and because I have a live screen, I'm going to show you. Wait, I'm going to show you an image of what we saw when the electron gun. Oh, I'm sorry. So there's a lot of speculation on what that color could be. Some of that coloration is the product of radiation and or radiation products that are interacting with material that is endogenous from Europa. In other words, material that is coming from the underlying ocean that's getting incorporated in the ice, that when it gets moved to the surface is hit with radiation. There's also a significant amount of material that is on Europa's surface, some of which might be coloring that coloration, which is deposited from Io. Io is a volcanic moon that injects material, including lots of sulfur-containing material, into space. That material actually is deposited, some of that material, on Europa's surface. Through radiation process, it's converted to sulfuric acid. By the way, terrible thing you want in contact with a biosignature. The third reason that you could get coloration of the ice, and what I was going to pull up is when we pulled one of the E-beam experiments out a few weeks ago, when they took it off a vacuum and removed the nipple from the manifold, the surface of the ice was pink. And that was what I learned is a feature that's been known all along. The electron beam knocks, okay, you got me on the spot here. The electron beam is knocking electrons out of salts that fluoresce. Yeah, I'm going to have to punt on this one because I need to refresh my memory on how this works. But to answer your question, there are a number of mechanisms that could account for that. Those could be organics. Those could be hydrated sulfate species. They might also be an effect of the radiation itself, which imparts a color to the salt ice mixtures.
Okay. This is Phyllis. I would point out that that's it? Okay, no questions. That the Earth has experienced at least twice, perhaps 3 times, an ice-covered ocean. As recently as perhaps 750 million years ago. Is there some way where we can detect the metabolic byproducts that would be embedded in the rock, the sediments of the ocean at that time?
First of all, you're totally right. Like if you would have visited the Earth 650 million years ago, it would have looked a lot like Europa. Depending on what proponent of the snowball Earth you are, either the soft or hard version. But with respect to your question, are we able, the answer is, I think the answer is yes, in that we are able to go back to those timeframes and look in the geological record to understand a lot about what what biological processes were occurring. So one thing I can tell you is that we know that when some of the oceans were covered with ice during these periods, that they went anoxic, because when the ocean goes anoxic, it allows iron to become widely distributed in the ocean. When ferric iron is in oxygenated ocean water, it becomes oxidized, and oxidized iron, which is rust, falls to the bottom of the ocean. So we know this because when the last snowball Earth terminated, it was associated with a banded iron formation. So while we're not able to look at that necessarily, I mean, the metabolic products there are not producing oxygen. So I think that probably qualifies. I think maybe you were asking me more specifically about how we might, would there be sort of molecular fossils of life from those periods, hundreds of millions of years ago in the like those are actually, that's sort of pushing the time frame on what we think those kinds of molecules could survive. Now, that's under Earth conditions. If you put those molecules at 100 Kelvin in ice on Europa, that might be a possible time frame that they could be preserved.
Yeah, well, some people are speculating, or perhaps they have some evidence that multicellular life actually evolved during that period of time under those conditions.
Yeah, that's right. Right after it, for sure. Yeah.
Yeah. Anyway, more comments, questions? Well, Andrew is always full of questions.
I have another question here. Can you comment on the mobility of the surface ice on Europa? Do we have enough photos over enough length of time to be able to see whether or not that ice is moving? And secondly, can you comment on the heat sources within the ice shell? Are there volcanic vents that we can detect even through the ice by a slightly higher, temperature in that particular spot, or any other way that we can know what the heat elements are within that ice shell?
Yeah, heat elements. I'm sorry, sir, I got caught up on what was the first thing? I'll remember it as soon as you say. The first part of your question?
I've forgotten. Let me remember it. Answer the second one, please, and then I'll remember the first one in a second.
Heat sources. Okay. Let me first say that I don't believe I said this. In Antarctic subglacial lakes, the reason that there are lakes there is because the ice at the base of the ice sheet is at the pressure melting point. Now, the surface of the ice can be at -50. The reason that the ice at the base is warm is because there's a flux of geothermal heat from the Earth. So even if you're in a place where it's frightfully cold at the surface, if you put enough ice on top of the Earth, it's an effective it's an effective insulating blanket. It traps that heat. And if the ice is thick enough, it means that you can actually trap that heat and start melting ice at the base. So in the case of Antarctica, the reason that there are lakes at its base, it's not because of global warming or anything like that. It's because of geothermal heat flux. That flux that we see on a place like the Earth, you would not expect. The geothermal heat flux on a place the size of these moons, all of which are smaller than the Earth, is much smaller. In fact, it would not be expected to be enough to actually maintain an ocean. So the heat that comes from these, it all have to do with the fact that these moons are in orbit around gas giants that stress them incredibly through tidal flexing, and that is the mechanism that is generating the heat that keeps the liquid water. I remember the first part of your question. You were asking me about how the rate of how these things move on the surface.
Yeah.
And the answer to that is we don't, because the first group of data we had from that are from Galileo, which is long gone, and It's an interesting question, because I'm not even sure if in the lifetime of the Clipper mission that they're going to be able to do that, but they should be able to re-look at sites multiple times to see if there were, I'm not really sure if anyone expects those kinds of dynamics in the lifetime of the mission, but it's something possible that we might know of in the decades that follow. What you can't do is point the Hubble telescope at Europa and see these kinds of features. So unfortunately, you can't get those kinds of long-term measurements from Earth.
So I had the impression, not from reading other material in our sources at home, but from something you said, that Cassini had already gone through the Enceladus plumes. So we actually do have data.
And I mean, the people who put that mission together didn't even know there's an ocean on Europa when they did that. We didn't even know there's an ocean there when it launched. And amazingly, this is NASA for you, right? Amazingly, those data have been so invaluable that, so you're right, we've done it, but we've not really, if at that point we knew that the instrument was going to go through an ice plume and it was going to look for signatures of life, there would have been slightly different instrumentation put on it. A mass spec is not a mass spec. There are like 100,000 different versions of mass specs, but yeah, you're right. It is actually the only ocean world that we have that kind of data from, direct data. When we get spectral data from Europa's composition, like what we're really probably getting information from is maybe like the top micron ice on the surface. We don't get spectral data that penetrate deep into the surface. What we think we are getting from the plume of Enceladus is material that is coming directly from the ocean. A paper published a few months ago thinks that actually it might not directly be the ocean. It might be pools of water that in the ice shell. Regardless, it would be material that was at some point associated with the ocean.
Right. So one more. Well, from me, I'm the one with the microphone. When would you suppose that the Clipper will take off?
Clipper launched last year.
Oh, it did?
It launched last year. Last summer, it flew in February. It flew past Mars. After it launched, it has 100-meter. It had a 100-meter antenna that had to be unfolded to do the radar measurements when it got there. And it's the kind of thing that, okay, it folds, all indicators are that it unfolded correctly. Is it really working? And in February, when it passed over Mars, it actually pointed that radar at I think it's the southern ice cap, and just basically did a test of the system. It didn't really collect any earth-shattering data or data that hasn't been collected before, but it showed that it's working. And we are 4 years, I think, from it to actually arriving into the Jupiter system, and then it will insert itself into this complex orbiting pattern where for 5 or 6 years it will increasingly make riskier flybys of Europa. And what I mean by that is later in the mission, I think if I'm remembering correctly, some of those flybys will be as close as 30 kilometers to the surface. Is that right? It's like that. It's something like it's distances that would make everyone super nervous very early in the mission, but later in the mission, they are willing to dive in that closely. So if there's a plume like you see on the artist rendition on my screen here, it could be the point of the mission where we know where those are, and it could target right into them. And then it will, that mission will be terminated by flying into Jupiter. And the reason for that is a concept called planetary protection. If This Europa is viewed as a special area that we would not want to contaminate with microbes from Earth. That's at least all the things I just told you would make it unlikely that if you crashed anything on the surface, anything could survive or live. Nevertheless, NASA will when that spacecraft is still controllable, it will dive into Jupiter and be terminated for the sole purpose of protecting Europa.
My question a moment ago about that mobility of the ice on the surface was, if you could find spots that are mobile, you could look at the edges of those mobile plates or spots and perhaps find places where the ice had caved away, choked off, and you might be able to find younger ice there that you could probe in some way. That was
that's right, and you've hit on it. The young ice would the young fresh material. That's the most desirable. Regions like that. If you couldn't catch a plume, some of that material is jetted into space. The larger grain size material will actually deposit on the surface. So there could be, regions where this stuff accumulates. Maybe not like glacial ice, but at least like in snowfields. Those would be super desirable regions to probe.
Thank you so much, Professor Christner. This has been fascinating, and I can understand your excitement and the energy that you are putting into these projects. Thank you so much.
Well, thank you. Thanks for all the great questions, and I hope you enjoy the rest of your specialty series. I'm super happy to be a part of it.
Thank you so much.
All right. Bye, y'all.